Click for IB Biology. We're looking at the prokaryotic structure. These, of course, are the bacteria. There is a blank PDF of my presentation down in the description that you can use to annotate with your own notes as we go through. There's a quiz, 10 questions with answers for you to check your own understanding. So let's get into prokaryotic structure. So these are the earliest of life forms noted in the fossil record in stromatolites somewhere in the region of 3.8 billion years ago, that this type of structure was already present. The key feature, which is best remembered about any prokaryotic structure, is that there is no nucleus. So there's no discernible nucleus within the cellular structure of a bacterial cell. Instead, they have what's called a nucleoid. Now this is like a piece of DNA, but instead of being a linear molecule, it loops around and reconnects. And so there's a loop of DNA, which is the nucleoid, or the chromosomal DNA, nucleoid. For some reason, some people call this a circle of DNA, which it certainly isn't a circle. It's often a loop of DNA. And on that are all the bacterial chromosomes. What is circular, or more circular, are very tiny pieces of DNA, which are features called plasmids. And these tend to only have a handful of genes on them, perhaps only 2 or 3. And interestingly, they can be passed from one bacterial cell to another in a process called conjugation. The plasmids become very important when we start looking at aspects of biotechnology. Other features within the cytoplasm of a bacterial cell, well, there's the nucleoid we've already mentioned, and you'll see plasmids, so rings of DNA, very small, but also you'll see the granular structure of the ribosome, where protein synthesis takes place. That's where they build proteins. Now, these ribosomes are different to the ribosomes in your cells and indeed of every other group. So prokaryotic ribosomes are measured at what we call 70S Svelberg units, which makes them smaller than we find in eukaryotic organisms. So there's no other organelles within the cell membrane. Around the outside of the bacterial cell, however, we do have elaborate wall structures, sometimes two or three layers to this, and it's typically formed what's called a peptoglycan structure. So it's not cellulose or chitin like in plants and fungi. This is made of peptoglycan, so that's another feature for you. The size of a bacteria is typically less than 1 micrometer, so it's less than 1/1000th of a millimeter in size. If you just try to visualize that, you will notice in the diagram that in the wall there are structures called pilus or pili. Now these are usually used as a means of attachment to the surface, or indeed the host, in which the bacterial cell is residing. So that could be attaching to the wall of the gut or something like that. Then there's also this crazy long structure here, which is known as the flagellum, which is quite different to that of a eukaryotic animal cell. The structure is very different. So I've just a brief look at this. The details of this you are not required to know, but flagella are used for mobility. They create propulsion, they move the cell forward. And the interesting thing about how this flagella does this is in a spiraling action. So it's not a simple up and down wave, but a spiraling action as well. And this spiraling action is created by a hook structure at the base of the flagellum, which I'm showing here in yellow. And that inserts into a sleeve that takes it through the outer wall of the bacterial cell, often the slime capsule, into the peptoglycan cell wall structure, and then it goes through to the motor unit, which is held within the plasma membrane, and that's where the mobility is generated in an ATP consuming process that creates this not only up and down motion, but a spiraling action that pushes the bacterial cell in this direction like this, and it's usually actually used moving towards chemicals to which it's attracted in a form of what is called chemotaxis. Bacterial cells, prokaryotic cells, often have unique shapes to their structure. Some of the classic ones are, for instance, the circular coccus shapes here. These, when they form straight chains like this is known as streptococcus. That's the one that causes you to have a sore throat, as opposed to staphylococcus. Here's staphylococcus here, which is forming more what are thought of as being more grape-like bunches of bacterial cells. These are some of the flesh-eating bacteria that we associate with infection to wounds. So streptococcus, staphylococcus, there's the spiraling forms here, the spirochetal type forms, Vibrio here, famous for causing cholera, and so on. So perhaps of the two kinds that we're particularly interested in is the bacillus, the classic E. coli type shape, which is kind of a simple rod type shape like this, and we have the staphylococcus, the aggregates of cells which hang together in our staphylococcus. Oh, sorry, not that one, this one here. Staphylococcus is here like this. Bacteria can also be distinguished in the way in which they react to stains, and the stain reaction places them in two groups, which are known as the gram-positive, and then there are the so-called gram-negative bacteria. Gram is referring to the staining technique. And within this, what we have is that, of course, both types of bacteria both have, in black here, the plasma membrane, but then the gram-positive cells have this very thick gram-positive peptidoglycan wall, whereas in the gram-negative bacteria, the peptidoglycan just forms this relatively thin wall here, and then the space created beyond the plasma membrane here and then to the outside is known as the periplasmic space, and in the gram-negative bacteria they have this additional layer, which is the lipopolysaccharide layer on the outside. So gram-positive, 2 layers, plasma membrane and peptidoglycan. Gram-negative, 3 layers. You've got the plasma membrane, the peptidoglycan, and the lipopolysaccharide layer on the outside. These staining up differently and allow the rapid identification of them with the gram technique. So after that quick look at the structure of prokaryotic cells, we're now going on to look at the internal structure of eukaryotic cells using electron microscopy. The link to the next video is showing there. There's a link to the channel so you can subscribe, and the playlist videos are linked on the right hand side.